Unit 3.2, Overview of Eukaryotic Cell Structure. If you remember, last week I compared the simple prokaryotic cell to Ole's Waffle Shop, saying that the gentleman who owns Ole's Waffle Shop is responsible for all of the functions that take place within the waffle shop. The same way, a prokaryotic cell has very little in the way of complex cellular structures. However, it is still able to carry out all the same metabolic, or many of the same metabolic functions of its larger relative, the eukaryotic cell. So this week I bring you Perkins, the analogy to the eukaryotic cell. Perkins is made up of many departments. Each one is responsible for one aspect of the operation of the restaurant. So there's human resources, there's a baking department, etc., etc. And you can think of each of those sections of Perkins as organelles in the eukaryotic cell. Eukaryotic cells are much more complex than prokaryotic cells, and each type of eukaryotic cell and each organism that is a eukaryote has certain physical characteristics unique to their particular cells. So that the structures that we're talking about tonight that pertain to eukaryotic cells may not pertain to all eukaryotic cells. Let's look for a moment at Table 5.1. These are the eukaryotic organisms that we will be studying in our microbiology class. First, we are going to see the different cellular compositions of the organism, and you will see that protozoa are always single-celled organisms. Single cell and unicellular are refer to the same kind of organism. Fungi and algae may be unicellular, but there are also multicellular fungi and algae. And then the helminths, which, as you remember, are the parasitic worms, they are multicellular organisms except for their reproductive stages. And the reproductive stages would be the unicellular egg or the larvae form of the helminth. The helminth, or the parasitic worm, has a very complex life cycle. So you may remember last week that on the whiteboard I drew a diagram of all of the different parts of a prokaryotic cell, and then as I referred to each part of the cell, I pointed it out on the diagram. If you'd like to open your book to page 125, there is a image of a gigantic composite of a eukaryotic cell, and that might be handy for you to look at as we're talking. But just below that is a diagram that outlines the cell structures in a eukaryotic cell. We won't be talking about all of those, but you may like to have that open next to you while we're moving through this lecture so that you can find your place within the lecture. You'll be able to refer to this table as we move along. Let's begin our discussion by talking about the cell envelope, the boundary layer of the eukaryotic cell. As you remember, the cell envelope is also the boundary layer of a prokaryotic cell. There are a few differences. Within the cell envelope is the external structure. It's the wrapping of the eukaryotic cell. It is composed of several parts. Some eukaryotes do have a cell wall. Some eukaryotes do not have a cell wall. We'll talk a little bit more in detail over the next few slides about which of the eukaryotes do have cell walls and which do not. But one thing that all eukaryotes do, in fact all cells do have, is a cell membrane. Just like the prokaryotes, the cell wall, if the eukaryote has one, and the cell membrane work together. Remember that coconut analogy of the stacked layers of the cell wall and the cell membrane? And that applies to eukaryotic cells. So even though those layers have distinct functions, they do act as a single unit to maintain the integrity of the eukaryotic cell. Referring specifically now to the cell wall, the groups of eukaryotes that have a cell wall are plants, fungi, and most algae. You have pathogens of the week this week that include some algae, and you remember if you've read about them already that there are some algae that cause harmful blooms, harmful growths that give off toxins that can damage both humans and other animals as well. The cell wall functions to give support to a eukaryotic cell and to give shape to the cell as well. Its structure is very rigid. Then the rigidity is due to the kinds of proteins that are built into the cell wall. And those kinds of proteins depend on the kind of eukaryote. Here are a couple examples here in your notes. Fungi have a protein called chitin in their cell walls, and algae have a protein called cellulose in their cell walls. And again, those algae are organisms that you studied this week in your pathogens of the week. So the big takeaway here from this slide is that not all eukaryotes have a cell wall. You know here the list of the types of eukaryotes that do have a cell wall, and you know which two examples of proteins that are included in the structures of cell walls to help and keep them rigid. Speaking now about the cell membrane, all eukaryotes, in fact all cells, have a cell membrane. Eukaryotes that have cell membranes but not cell walls are as follows: the protozoa, a few algae, and all animal cells. Now if you remember in your pathogens of the week, you studied one of the protozoa, and that was Entamoeba histolytica. In the cell membrane, there are functions and aspects to eukaryotic cells that are similar to those of prokaryotic cells. The cell membranes of both types of cells are responsible for transport of nutrients into the cell and wastes out of the cell through the selectively permeable membrane. Both eukaryotic and prokaryotic cells use cell membranes to maintain their cellular structure, their cellular shape. Now this is very important for eukaryotic cells that do not have a cell wall, because you can imagine that cell membrane is the outermost covering, and that cell membrane is responsible for holding in everything that's inside of a eukaryotic cell that does not have a cell wall. An aspect that eukaryotes have about their cell membrane that is different from those of prokaryotes is that eukaryotic cell membranes have membrane-bound organelles, and these organelles can take up to 60 to 80% of the volume of the cell. In one way, the chemical composition of the cell membrane of both prokaryotic and eukaryotic cells is similar. They both have a phospholipid bilayer that has proteins embedded in it. And then, unique to eukaryotes is the fact that the cell membranes of eukaryotes need extra strength. Remember, they have no cell wall, and that extra strength is imparted by proteins called sterols. And also, unique to eukaryotes is the fact that carbohydrates are present in the cell membrane. Now you can see a little asterisk there that refers you down to the bottom of the slide, just as a reminder that there is one group of prokaryotic cells, the mycoplasma, that do not have a cell wall, and you studied those last week. In fact, they were one of your pathogens of the week. So among prokaryotes, mycoplasma are pretty rare because their membranes also contain sterols. Moving on now to a variety of external cell structures that a particular eukaryote may or may not have, depending on the cell. We are going to begin with locomotor appendages, including the flagellum and the cilia. And you can see some pictures of a flagellum, and you can see some pictures of cilia on one of the organisms on this slide, and we're going to look at each of those external cell structures in detail on the next few slides. Even though the locomotor appendage flagellum has the same name in a prokaryote and in a eukaryote, there are many differences between prokaryotic and eukaryotic flagella, and both in the structure on the outside of the flagella and on the structure on the inside of the flagella. Eukaryotic organisms that have flagella include some protozoa, algae, a very few fungi, and actually some animals. These would be very small animals, but there are animals that have flagellum. Unique or not unique is the fact that the protein flagellin is found in both prokaryotic and eukaryotic flagella. I don't think you can do it from this YouTube recording, but if you go back to your original PowerPoint or your original PDF or to the link in D2L, you will be able to watch a few seconds of a video of some Trichomonas vaginalis organisms that have a flagellum, and you'll be able to see the little flagellum wiggling around. These flagella help the single-celled Trichomonas organism to navigate through moist conditions. Trichomonas vaginalis is a single-celled organism that is responsible for a type of venereal disease. The picture on this slide shows you a cross-section of a flagella, and inside of this flagella you can see little tiny rings arranged in a certain pattern. Those little rings are called microtubules. So I just want to make sure you understand what a cross-section is. If you went to the Wise Markets and you asked for some slices of cheese, they would bring out a big log of cheese and put it on the slicing machine, and then they would give you slices of cheese. Well, this is the flagella is a long, thin cell structure. So think of cutting a slice off of that flagellum, a very thin slice, and then lying it flat under a microscope. That would be considered to be a cross-section, and that's what you're looking at here. So, although the flagellum is not unique to eukaryotes, its structure is unique. It is not the same structure that you find in bacterial flagella. So there are 9 pair of microtubules, those little rings, 9 pair of them, and then 2 single microtubules in the middle. And that's referred to as a 9+2 arrangement of microtubules. Those microtubules are responsible for the movement of the flagella. And if you do take the opportunity to look at the Trichomonas vaginalis organisms moving around, you'll see how those flagella are responsible for whipping the organism around. Another unique feature of eukaryotic flagella is that the flagella themselves are covered by an extension of the cell membrane. So think back for a minute about the flagella of bacteria. They have the long whip-like portion of the flagella, and then the base of the flagellum is anchored in the cell wall, cell membrane complex. But there is no covering on that flagella. It's out there in the open, exposed to the elements. Now think about a eukaryotic flagellum. And I'm going to make an analogy here. Imagine that you yourself, your body, is a eukaryotic cell, and that your arms and your legs are flagella. Now, imagine you without your layer of skin. You would have your flagella, sort of an integral part of your body. Now, put your skin back on, and think of that skin as the cell membrane of an individual eukaryotic cell. So that cell membrane, or in your case, your skin, is covering both the flagella and the body of the eukaryotic cell. And if you need a more detailed explanation of that, you could refer to your text. Another unique aspect of eukaryotic flagella is the fact that they are 10 times thicker than the flagella of prokaryotic cells. So even for tiny eukaryotic cells, their flagella are quite a bit thicker than those of the prokaryotic cells. A second external structure of eukaryotic cells are cilia. Now, we did not talk about cilia when we talked about prokaryotic cells. Prokaryotic cells do not have cilia. Cilia are found on a few protozoa and a few animal cells. As far as their structural components, how they're made, they are similar to flagella. However, they are shorter and much more numerous. They function to help some eukaryotic cells move around, but they also function to direct nutrients and tiny food particles towards the actual cell and sometimes into the cell itself. The image that you have here on this slide is of a very tiny single-celled organism that has a little slit in it that's called an oral groove, and that slit has cilia that surround it, and the cilia sort of act as little fingers, little waves to try to push the food and the nutrients into the oral groove of the organism. The cilia are also sometimes important in filtering, and that means they are important in preventing certain particles from entering into eukaryotic cells. If you would like to see some cilia in action, click on the link in D2L or click on the PDF of this unit in our D2L website, and you can watch some rotifer cilia in action. And when you see those rotifer, you'll see that their cilia are placed differently than the cilia in the image of this organism on this page. So we have to remember that the number of cilia, the size of the cilia, the function of the cilia is dependent on the organism. Another structure that some eukaryotic cells have is the glycocalyx. For those eukaryotes that do have a glycocalyx, it's the outermost boundary, which means that it surrounds the cell membrane of those eukaryotes, and it is in contact with the environment. Groups of eukaryotic cells that have a glycocalyx are all within the same group of eukaryotic cells that do not have a cell wall. So if an organism has a cell wall, it does not have a glycocalyx. If it is without a cell wall, it may have a glycocalyx. Eukaryotes have 3 different types of glycocalyxes depending on the organism. The glycocalyx could be a network of fibers, it could be a slime layer, or it could be a capsule. The glycocalyx is made of polysaccharides, and it has the same functions as the prokaryotic glycocalyx. It protects an organism from getting dehydrated, it keeps nutrients in, and it is sticky. Remember, the glycocalyx of prokaryotes is quite sticky, and that stickiness of the glycocalyx allows it to adhere to or stick to surfaces. We're going to discuss the internal structures of eukaryotic cells now. These internal structures are considered organelles. Each organelle has specific functions. The organelles are located within the cytoplasm. And when I say that, you have to remember I also said that many of them are bound to the inside of the cell membrane, and that's true, but the inside of the cell membrane comes up against the cytoplasm. So even organelles that are bound to the cell membrane also protrude into the cytoplasm of the cell. Below, at the very bottom of this slide, you can see a list of the organelles that we cover in this course, and there are other organelles that we won't be discussing in this course. We will talk about each of the organelles listed here on this slide over the next few slides. Every eukaryotic cell has a nucleus. You could think of the nucleus as the control center of the cell. It is the most prominent organelle. It's when you're looking at a eukaryotic cell under the microscope, it's the nucleus that really stands out for you. The image here of the slide that you're looking at has a photograph of a cell. That's represented by that circular shape, and then the very dark, very large area in the center is the nucleus of the cell. If you look at the picture underneath, that's a drawing of a nucleus, you'll see that the nucleus is kind of like a shell, and that nuclear membrane is what forms the shell around the cytoplasm or the chromosomes and the nucleolus, which is located within the nucleus. And then just to the left of that purple nucleus, you'll see a blown up portion of that image, and you'll see that the nuclear membrane isn't just a single layer, it's actually 2 layers with some space between the layers and some holes that are on one layer and on the other layer, and it is through the pores, the nuclear pores, that nutrients pass in, wastes pass out. So that nuclear envelope or nuclear membrane surrounds the nucleus. It has parallel membranes. It's perforated with pores. We talked about the function of the pores for the passage of molecules in and out of the nucleus. You can see, again, let's go back to that little purple diagram. You can see the nucleolus, the dark purple bundle there inside the nucleus, and it is within the nucleolus that ribosomal RNA is made. The chromosomes are also located within the nucleus, although they are not visible in the purple drawing that you see on this slide. Most eukaryotic cells have internal structures called vesicles. We're going to talk about 3 kinds of vesicles tonight, and we'll doing that one at a time. The first one is the lysosome. You can see the image on the right, and the red arrow is pointing towards a small lysosome located within the cytoplasm of that eukaryotic cell. Within the lysosome are enzymes that are responsible for digesting food particles and other nutrients. They are also responsible for the digestion or destruction of microorganisms that happen to invade the eukaryotic cell. A second eukaryotic structure that is typically becomes internal to the eukaryotic cell is the vesicle. Is the vacuole. And a vacuole forms because a portion of the cell membrane makes a little pocket around a particle that at first was exterior to the cell. So if you look at the diagram, you'll see a red arrow pointing towards a few little particles that are hovering above that eukaryotic cell. Those would be some particles that need to be digested, and you can see the cell membrane is reaching out to envelop those particles, and once that envelopment has taken place, the eukaryotic cell now has an internal vacuole within its cytoplasm, and that vacuole contains the food particles, or it could be a microorganism, that have been captured by the cell and taken inside. Now, in the text it says that the vacuoles contain particles to be digested, and that's what we just talked about. They can also contain particles to be stored, or once a particle has been digested, any waste that results from that digestion is then held within the vacuole to be excreted by the cell. The final cell vesicle that we're going to talk about is the phagosome. PHAG, or phage, or phage, refers to eating or digestion. So when a vacuole containing some particulate matter or some liquid nutrient merges with a lysosome, which contains the enzymes to digest or destroy that particle in the vacuole, a phagosome is formed. So the phagosome is the merger of the vacuole and the lysosome. Another cell structure that all eukaryotic cells have is the mitochondria. And you probably remember from every biology class you've ever had, at least it was really memorable for me, when teachers referred to the mitochondria as the powerhouse of the cell. And that's really true, because the function of the mitochondria is to produce the energy for the cell in the form of ATP. And there are some characteristics of mitochondria that are unique to the mitochondria, and different from all the rest of the characteristics of the eukaryotic cell that contains those mitochondria. Well, let's talk about that for a minute. Mitochondria divide independently of the cell that contains them. As you know, when a cell reproduces, one way for it to do that is to divide from one cell into two cells. And when that happens, all of the internal structures are duplicated so that from the one mother cell, two daughter cells are formed with all of the same internal structures. However, mitochondria can divide at any time within a cell, independently of the time when the cell itself is going to divide. Something else that's different about the mitochondria compared to other characteristics of the eukaryotic cell is that mitochondria contains its own DNA. Remember, most of the DNA of the eukaryotic cell is contained in the nucleus, but mitochondria has its very own DNA. It also has ribosomes, and remember that prokaryotic ribosomes have a size of 70S. You're going to learn on a few slides from now that eukaryotic ribosomes have a size of 80S, except for the ribosomes in the mitochondria. Ribosomes in the mitochondria are 70S, the same size as mitochondria in bacteria. Some eukaryotic cells have an internal structure called a chloroplast. Chloroplasts can be found in algae, and they can be found in plants. Chloroplasts are similar to mitochondria in their shape and some of their internal contents, but they're typically larger than mitochondria. Chloroplasts contain special pigments, and not only do they vary in size from the mitochondria, but they also can vary in shape from one type of chloroplast to the next. Chloroplasts function in energy production, but they do it differently than mitochondria. Chloroplasts make their energy for their cells by photosynthesis, and that is the conversion of sunlight into energy. One of the byproducts of photosynthesis is oxygen. So oxygen, of course, is very important to other living things. The chlorophyll is the name for the green pigment that traps that light energy, which is then converted into energy for the cell. But chlorophyll is not the only kind of pigment that can be found in a chloroplast. Depending on the eukaryotic cell, there may be other pigments present, including the carotenoids. If you think about a carrot, think of that word carotenoids, it's very similar, isn't it? The carotenoids are an orange-red pigment that is found in many algae and plants, and it is the pigment that gives the color to carrots. On the original PDF of this unit or on D2L, there is a link to a little article about the blood red water that can be seen in this particular lake in Turkey. And it's a very short little piece, but I think it's very interesting. And as you know, the harmful algae bloom is one thing that you did study or will be studying in your pathogens of the week 3. So this is a reference with a little bit more information about harmful algae blooms and the responsibility of chloroplasts in those blooms. Another internal structure of all eukaryotic cells is the ribosome. Now don't be fooled by this picture of the mitochondria. Look a little more closely and you'll see that the tiny little blue dots within the mitochondria represent the mitochondrial ribosomes. Then look at the image here on the bottom. That purple sphere that you see is a small part of a nucleus. The enfolded layers there represent the rough endoplasmic reticulum that surrounds the nucleus, and the little specks, the little blue specks on that rough endoplasmic reticulum represent eukaryotic ribosomes. So eukaryotes have two sizes of ribosomes, 70S ribosomes in the mitochondria. Remember, these 70S ribosomes are the same size as ribosomes found in prokaryotic cells. The eukaryotes also have 80S sized ribosomes attached to that rough endoplasmic reticulum and also floating around freely in the cytoplasm. The function of the ribosomes in a eukaryotic cell is the same as that in a prokaryotic cell, and that is to synthesize protein. In looking more closely at the cytoskeleton in this diagram, you can see that the cytoskeleton itself is made up of both microfilaments and microtubules. Microfilaments, if you direct your attention down to the image itself, you see that fat blue star with some arrows pointing towards the microfilaments. They are made of a protein called actin, and the microfilaments are what aid in the helping the flow of cytoplasm through the cell and those amoeboid movements that we talked about on the previous slide. Look again at the diagram at the fat red star, and you will see that that arrow is pointing to some long hollow tubes called microtubules that function to maintain the shape of the eukaryotic cells, and that's really important because remember most eukaryotic cells don't have cell walls. The microtubules also help in the movement of the flagella and the cilia of eukaryotic cells that have those cell structures. Here's another view of the cytoskeleton, and this is a photomicrograph, so this is an actual photograph of a cell and its cytoskeleton. You can see the microfilaments in orange and the microtubules in green. Interestingly, the nucleus is in blue, and it looks like the cell has two nuclei, doesn't it? That's possible, but it's more likely that that nucleus is a C-shaped or an S-shaped nucleus, and that this cross-section of the cell has sliced through both ends of the letter C, so you're looking at just two different ends of the same nucleus. Now this is a composite image of a eukaryotic cell. It's the same one that's in your textbook. And what a composite image means is that this is the dream eukaryotic cell with every single possible organelle known to man within it. Not every eukaryotic cell has all these parts, but this composite cell does, and you may find it useful in studying to be able to put a place in space to all of these different components that we've been talking about. And this last slide isn't so helpful to us here as you're listening to this recorded lecture, but it is also represented in the PDF of this lecture that's on D2L, and it gives you an outline of the structures that we talked about tonight. I drew a line through the few structures that we did not talk about, but you should recognize the names of all of those other structures, and these are things that you should be familiar with as we move through the course, because you will see that this information is going to build on itself. From one lecture to the next, we are going to learn more about the significance of each one of these structures and its place in metabolism, or the immune system, or the production of energy. And we have reached the end of Unit 3.2. So please email me or call me if you have questions. I'd be happy to go over any part of this presentation that you'd like me to, either by phone or just to point you to a place in the textbook that might give you some more information. So thank you for your attention, and you'll be hearing me again in Unit 3.3.